Thank you all for being here. My name is Chun-Yu Lin. I'm from the Institute of Bioinformatics System Biology in National Yangming Jiaotong University. And today I'm glad to have this opportunity to share this article we have published in the Nucleic Acid Research 2024, the gene set correlation enrichment analysis for interpreting and annotating gene print profile. And as you know, with the recent advancing of the sequencing technology, we have seen the cost go down and accuracy improve. So the key challenge isn't just to get the gene expression data or analytic data, but interpret it in a meaningful way. One of the most effective approach we use is the pathway or enrichment analysis methods. And based on the knowledge base, like the KEGG pathway or the gene ontology. So based on this method, we can access the pathway or the functional annotation significance. And up to now, more than, I think, around 100 enrichment analysis methods has been developed. And we can roughly to separate them into the non-topology based and the topology based approach. And the first generation non-TB method is the overrepresentation analysis called ORA. And ORA assesses whether the differential expressed gene in a given list are over or underrepresented in the gene set, like the pathway. And several many famous methods like the onto-express, geostate, or the David, and so on, depending on the ORA to design. But the ORA method disregards the meaningful change in gene expression. It means they're treating each gene equally. So to address this limitation, the second generation non-TB method called the functional class scoring, FCS. And the FCS assumes that weak but coordinated change in gene set, like the pathway, may also play the key role. So very famous one is the GC, GSA, PADOG, all depending on the FCS to design. But also similar to the GORA, they suffer a challenge is that they they failed to account for the crosstalk and like the overlap between the pathways. So it means they analyze pathway independently, so may let this limit their ability to describe and capture the biological phenomena. So the third generation, called the topology-based method, they try to address both issues to utilize the pathway or network knowledge as the background. Okay, so we can get the interaction between or dependency between the given gene list or between the given gene and the pathway to enhance the significant detection, association detection. So also have several methods like the SPEAR, longitudinal, NEA, and so on to design by that. However, there are several limitations for current topology-based method. The first issue is that how to select the background network for the different studies. And this is the issue is about mostly we choose the protein-protein interaction data as the background network, but protein-protein interaction data currently is incomplete. So one way we can do is that to collect all the data, maybe experimental identify from the different cell type, different state, but we don't have enough data, so we collect all of them to build the network as the background. But there is a problem will be happen is this network is aggregated one, not context-specific one, so it will lose the context-specific informations. So we want to address another unmet needs is about the prioritize about the DG or the gene list. Because for most of the user and the biologist, when they conduct the gene enrichment analysis method for their gene expression data or the DG list, they get a list of the enriched pathway, and they usually will ask another question is about which gene play the key role in impacting this pathway. So this gives us the idea is can we sort or rank the gene involved in the gene list or the gene expression data associated with as a pathway, we can based on their importance and to help the biologist or the user to guide the plan of the following experiments. So we propose the method we call the gene set correlation enrichment analysis, G-score. And G-score for the given gene impression data from the user, we focus on the cat sample to construct data set derived collaboration network. So the network is constructed by your given data. And then for each pathway and a collection of pathway, we will quantify a group of genes, it means the digit list, or individual genes based on the co-expression gene pair between the query. Query include the digit list or its individual members between the query and the certain pathway, the co-expression gene pair frequency. And also we can compare to all the pathway, the co-expression gene frequency among all the pathway as the background probability. So we can use the hypergeometric test to examine the significance, not only for the digit list, a list of digit, but also each individual ones. So we would like to benchmark different topology and non-topology methods, include our G-score. So we collect about 75 microarray data from GEO in 15 diseases, as well as the 10 RNA-seq data from the TCGA projects. And we follow previous work to define the target pathway for each gene set. The target pathway means that for each gene set, already know that it is for what kinds of disease or condition and has a corresponding KEGG pathway. The pathway for this gene set we call the target pathway. So for each data set, we conduct the different enrichment analysis method like our G-score based on the human or KEGG pathway. And then we can get their significance, and we rank their significance from the most significant to the less significant. And then the rank, the median of the rank among the 75 data set can be shown in the violin plot. So it means that the performance is based on rank the target pathway on top. And then the smaller rank value means the better performance. Comparison to other 10 methods, we can find that over the TB and the non-TB method, G-score and the PADOG achieve the best performance. It means the lowest median rank value, no matter the different expression platforms. And next, we would like to ask whether the DEG involved in the pathway could be significantly associated with the original pathway using the enrichment analysis method. So for each pathway, the DEG record in this pathway means the involved DEG can be considered as the positive one, but it belongs to another pathway, not in this pathway, can be considered as a negative one. And then please note that only the G-score and the NEA can query only one genes. So comparing to the NEA method, our G-score had a higher accuracy, precision, and lower false positive rate. Next, we further apply and validate the G-score and the other methods to annotate and interpret the gene impression data from the cell patient sample with the COVID-19 infection. And by applying this method on the 4 different data sets, we first compare the ability to identify the COVID-19 target pathway. Similarly, the G-score is the best able to detect the target pathway of the COVID-19 in comparison to the other method. Additionally, we collect the 17 COVID-19 related pathways defined by the KEGG, and we want to examine whether the COVID-19 related pathways are significant impact in a given phenotype. So the dot plot shows the significant association between these 70 COVID-19 related pathway and the DEG list derived from the 2 RNA-seq data set of clinical patient. And statistical analysis using the visual test show that our G-score can identify more COVID-19 related pathway than the other methods. And moreover, the heat map of significant association between the individual DEG and the COVID-19 related pathway show us about the DEG with a strong association across the multiple pathway are more likely to be the COVID-19 related gene, like the left panel, and the comparison to weak association in just one or specific COVID-19 pathway, suggesting that G-score can determine which of this DEG tend to be the COVID-19 related genes. Finally, we built a G-score web tool that the user can free to use. And user can upload their unimpression data set and also upload the collection gene set or select that gene set we already curated, like the KEGG, gene ontology, and so on. And then for many model organisms, we provide this service. And also we use the interactive table, dot plot, heat map, as well as the network graph to show our output results. And the take-home message I would like to let you can summarize about the G-score use the dataset-derived cooperation network to compensate for the limitation of the other method, and also help to integrate the annotated gene impression data, not only for the DG list, a group of DG, but also individual member in this list. And also the G-score online tool provide the easy-to-use framework based on the clear defined procedure, as well as the intuitive visualization of finding. And this is the URL for the G-score, or you can scan the QR code. Finally, I would like to thank all the co-authors in my labs, and also our collaborator, like the Professor Li Jiahua in Taipei Medical University, Professor Li Riqing from Tri-Service General Hospital, Professor Zheng Yuyao from Shenzhen University, and also thank you to founder who supports us, and thank you for your attention. Our poster ID is the 24X1. Please, I would like to take your question, or you can contact me via the email. Thank you.
Okay, I have a question. Does can G-score work for both the directed and undirected networks?
Oh, currently the G-score is depending on the cooperation network. I think it's maybe the undirected network.
Okay.
Yeah, but we try to integrate about the directed network. Just need to using the KEGG pathway or the rectactome pathway into the cooperation network. But currently, it's undergoing project, so not yet to try about the directed network. But we believe the directed network as the big one can improve the performance because it's more close to the pathway. Yeah.
Okay.
Thank you.
Any other questions?
Yes, so if I understood correctly, this method uses only the differentially expressed genes, right?
Yes.
So it does depend on the threshold, right? You're not using the expression of all the genes like the second generation method for enrichment.
You mean the, pardon, can you say again? You mean the, yeah, based on the gene expression, so for the question between the two genes across the CAS center to build the cooperation networks.
Any other questions, comments? No?